All right, today we are hearing about winter barley from a bunch of people, and we're excited to have them here. Welcome, everybody. Welcome, folks online. Reminder, as always, we're recording, so keep that in mind. Thank you to Forever Green's funders, Environment and Natural Resources Trust Fund, Minnesota Department of Agriculture, Clean Water, Land and Legacy Amendment, Builders Vision Philanthropies, and the McKnight Foundation, among other very generous folks that make this work possible.
So yeah, I'm Kevin Smith. I'm in agronomy and plant genetics. I do plant breeding, and one of the crops I work on is winter barley. So we're going to talk a little bit about some of the developments in winter barley. In particular, there's lots of folks that are working on this project in many different ways. But we're actually going to hear from Maicy today, who's subbing in for Waleed, who had a conflict. So she will present sort of a coming attractions that's going to relate to some upcoming research that is funded through the current or the new FGI grant program. And then Sydney is going to talk a little bit about a project that's related to that. And then I'll spend a little bit of time talking about some of the breeding progress that we've made. But I put this slide together because it kind of just helped me think about the whole wide range of work that has to be done to really do the developing winter barley for Minnesota cropping systems, right? So it's, obviously I'm working on much of the breeding and genetics side of things, but there are all sorts of other aspects to this work that are important that need to come together in order to make this whole system work. And so I kind of outlined them here, and we've talked about some of these things in the past. One of the things that I like to do with our little one-time slot per year is to bring in folks that are part of other parts of the dimension of this work. And so in the past, we've presented work on some of the agronomics that we've done in terms of optimizing a system. And the system we're thinking about is a double cropping system with soybeans. Jake Younger's group has talked about an actual double cropping experiment and also some nitrogen cycling work that's an important part of that double cropping system. Not just understanding the agronomics or optimizing the agronomics, but also making sure that we can measure whatever else we want to measure in terms of the environmental components of that system as well. And then, of course, the commercialization folks have talked about the ECO program, which includes winter barley, and they've also done some work with a large local maltster to try to get some actual winter barley contracted for malting, which would be a relatively novel new thing. And that's still a work in progress. And then there's a lot of end use work. We're fortunate in winter barley or barley in general that at least from the malting quality side of things, there's a USDA lab in Madison, Wisconsin that provides a fantastic service for all of the different breeding programs and research programs working on barley. And so they provide a lot of our quality data that we use to look at that. But there are other possible uses as well, feed, food. And so there are some efforts to look at some of those different uses and what would be needed to support those uses in, again, a cropping system, winter barley cropping systems. So that's just kind of the big picture. And as I said, Maicy will start out talking about this new project that has to do with winter survival. So just to remind you why we're talking about winter survival, if you look at this slide closely, this is what we observed last year in our trial. So these are just single row plots that we plant just to do winter survival. So we plant out hundreds or even a couple thousand some years of just different germplasm every year, multiple locations, just to measure survival. And for the previous 4 years, we've actually had pretty decent survival. We've gotten good data from our yield trials, but last year was quite different. So you can see there's this one little plot there over on the left that looked like it was green. So obviously took some careful notes on that one and a few others that had some green on them. So this was in St. Paul. Interestingly, we also do this exact same trial in Rosemont, and it's more in a little protected woodland area. And we actually got quite a bit more survival there. So even, small distances. So one thing we've learned is there's lots of locations. We do it in Crookston as well, which is almost guaranteed to kill many.
Yeah, my name is Maicy Vossen. I'm a current PhD student. I am co-advised with Waleed Sadek and Eric Watkins over in horticultural science. So I currently study freeze tolerance in perennial ryegrass, so a species of turf grass, but a lot of the stuff that I'm working on is very translatable to other species, and I'm going to kind of like talk about how those things are translating into this new project. But yeah, so this project that we have coming up is working on an integrated approach for improving freeze tolerance and winter survival of continuous living crops in Minnesota. As with all projects, we've identified some challenges that currently exist within the breeding world and these continuous living cover crops. So we know that breeding for freeze tolerance can sometimes have really slow progress and can be a bottleneck for really developing these crops that reliably overwinter every year. This is kind of due to several different factors, but a couple of them is due to the complexity and unreliability of field trials, the lack and underutilization of proxy traits, and the lack of envirotyping, specifically in Minnesota for this project. For this project, we have identified 9 different breeding programs for which freezing tolerance is really critical to develop. And within these programs, they're all at different levels of development. So that's also one of the challenges here as well. And they often operate mostly independently. So there's also some issues with that slowing down technology and knowledge transfer as well. So we're really working to kind of like integrate these with the goal of improving the efficiency and integration across these 9 breeding programs to work to fast track the freeze-tolerant germplasm. This project comes at this goal with different areas of expertise in breeding, physiology, phenomics, and envirotyping. So this is the list of the different crops and breeding programs that are going to be part of this project. So as you can see on the very right-hand side of this table, the number of genotypes to be screened for each crop is a very wide range. And so that really relates to how different these programs are in their development. But across the board, we are anticipating being able to screen almost 1,000 different genotypes for this project across 6 different functional types of plants. So we should be gaining some new information, also working on that freeze tolerance as well. So I'm just going to give like a very high level overview of each of the objectives. If there's other questions, we can ask at the end if there's time, or I don't know how to give my email, but like people can also like reach out to me or Waleed or Kevin with questions about the project as well. But for the first objective, as we kind of have alluded to, we know that for field trials to get reliable winter hardiness data, we need lots of years and lots of locations. And unfortunately, there's lots of inconsistencies with the environment, so that makes it really challenging. And also for some of these emerging breeding programs, it's not as feasible to have these large-scale field trials. So part of this objective is to create and implement a smaller scale controlled environmental assay that can be used to accelerate this screening. So I just kind of like put up 3 basic steps of how this works, but I don't know if people are familiar with like an LT50. It uses that as kind of a basis, but instead of screening at a lot of different temperatures, we're doing it at more of like a high tolerance, middle, and low tolerance temperatures to kind of just get an estimate for the tolerance of these different genotypes. So this will be really helpful for recently established breeding programs to just get the degree of freezing tolerance for new germplasm and identify genotypes that might be used as donors, and also just to inform the design of maybe the first field trials for some of these developing programs. And then for more established breeding programs, these will be really helpful to validate past field trials and evaluate new germplasm that's entering the breeding pipeline. And also, we'll be working to identify genetic markers associated with freeze tolerance for marker-assisted breeding in winter barley specifically. For the second objective, we know, and it's pretty well established, that freezing tolerance is associated with smaller metaxylem vessel diameters across a wide spectrum of species. So these are the water transporting tubes in the plants. And this wide spectrum of species includes, it's very well studied in winter and spring barley, also other monocots and some dicots as well. So it's pretty well established. And also we know that genotypes with smaller metaxylem vessel diameters tend to express higher hydraulic resistance to xylem water fluxes, which ultimately results in lower canopy transpiration rates. So that's something that can be measured as a functional proxy trait. But through this objective, we are also using candidate anatomical traits that can be measured through laser ablation tomography, which is what the images are showing. This is perennial ryegrass from my PhD work, but this is a much more high throughput way to get cross-section images of plant tissues compared to like traditional histology or other optical microscopy measurements. So we'll be working with these to really fast track that screening. And I think what's really cool about this objective is that it's using proxy traits that are constitutive. So they don't require any acclimation for actual evaluation of these traits. So we'll be able to measure this at any time during the year. And so that will really kind of speed up this process. Then the last objective, which we'll kind of get like a little bit more information on kind of how this stuff is done, and some of the work that's actively being done from Sydney. But so for this third objective, we know and acknowledge that identifying freeze-tolerant donors, validating these traits, and releasing freeze-tolerant varieties are obviously very necessary when working on winter hardiness, but it's not entirely sufficient to implement these new economically sustainable continuous living cover crops. So the goal of this objective is to really kind of create some decision support approaches for farmers to make informed decisions about what crops they're going to be bringing to their farms and their local environments. The reason that this is really important to understand the envirotypes of across Minnesota, create these risk maps to guide our decisions, is because in some locations, high freeze tolerance is not always required. If you get lots of snow cover very regularly and reliably every year, you don't need as much freeze tolerance in that specific location. But if you're not getting that snow cover, you get really cold temperatures very regularly, then you need to adjust in what you're actually deciding to grow. And this is also really helpful to look at not only winter survival, but also yield trials in this objective, because breeding for freeze tolerance, that it might have some physiological trade-offs, and that could limit potential for yield. So cool season grasses like winter barley can incur some yield penalties if there are smaller metaxylem vessels in the plant, because it might have a lower ability to mitigate heat stress because there's not as much canopy transpirational cooling that's happening. This same vascular phenotype can lead to improved yield performance in environments that are really dry, because there's water conservation that happens because of the same smaller xylem vessel diameter. So it's really important to kind of like understand the envirotypes that exist across Minnesota, so we can make sure that we're giving everyone the support that they need to choose the correct continuous living cover crop.
Are you able to look at fatty acid profiles too? Like sometimes like that can be a factor in freeze tolerance?
Yes, it definitely is. It's not a specific objective of this project, but I do know that there is work happening at the university related to that. I've done some work related to fatty acid composition through my own PhD work, but it's not a direct objective of this project, but it's definitely super important.
You mentioned some possible anatomic traits that are proxies for the metaxylem side. What might those be, and could any of them be measured in the field in a high throughput system? Yeah,
so as like a good proxy that we're seeing like well correlates is the transpiration. So specifically in environments where there's like a high transpirational or like a high VPD, where there's like a evaporative demand, it really segregates. And so you know, if you want to get out there with a live core, that could be something that could be measured in the field.
We're talking more height.
Yeah. So, specifically for this project, the method that we'll be using to evaluate transpiration as a proxy happens in controlled environments using like a gravimetric approach. So measuring how much weight of water is lost due to canopy transpiration. But in the field, I know I'm working kind of with this a little bit, but it's not exactly great. But using like thermal cameras to see the like canopy cooling, that might be something that's more feasible. But again, there's some challenges. If you have like a really nice cool day, you're probably not going to see any differences. So yeah, there's lots of things that maybe could work, but yeah.
I had a question about how you're testing the different tolerance levels.
Yeah.
Because it seems like they're, back to this moisture question, like there's the, the temperature itself, there's the duration of cold temperature, whether it's wet or dry in the soil. So are you layering on like a dry, cold winter or just a cold winter? Like, how are you kind of wrapping your mind around all the complexities of what it means to be cold tolerant?
Yes. Yeah. So the screening method that we're talking about for objective 1, it's just using a very like standardized protocol across the board. We're not adding in a lot of environmental variables. However, we have seen good correlation to winter survival using this method. So yes, it doesn't take all the extra complexities into place, if you have snow mold out in the field or, just different things that could happen, but it does give us a good starting place.
Is it possible to like take a sample of like a stem if there's branching? And it's not high throughput, but I don't know what that, I'm not familiar with laser ablation.
Yeah.
But would that be faster maybe than out there? And I mean, because you could save it for later.
Yeah.
Maybe you could evaluate in the winter time.
Yeah.
Is that something that could be done? How easy is laser ablation?
Yeah. So there's not a lot of facilities that have this kind of technology. So we have in the past, and for this project, are going to work with Penn State. They have like a core facility that they're able to, for several hundred samples, we can get a turnaround time of like a week or 2. So it's pretty high throughput in that sense. We don't have to do any sort of staining or extra like preservation of the tissues. You can just like take it from the field, put it in 70% ethanol, put it in the fridge, and then you just send it to them when you're ready. And then they do any like dehydrating sample prep and stuff there. But the process itself is also really cool because you can get some like 3D images as well. So it offers kind of some unique information that you can't really get from like traditional histology when using like a microtome and stuff like that. So
cool.
Yeah. I don't know if that exactly answered your question, but
I want the contact from Penn State.
yeah. They're really great. We have worked with them for several years, even before they became a core facility. So they're great people. Yeah.
I have a question then on like the actual analysis of the sections. Like, would you then have to go through for each species that you're sampling to like maybe an image to like a specific pipeline to do that analysis of the xylem?
Yes. So that will be probably part of this, is to really iron out that kind of protocol. It's a little bit challenging because like not every leaf looks exactly the same. And so to ask it to like choose this region, that creates some challenges, but I think it's probably feasible, especially because we'll have like a pretty large number of samples that are going through this.
Maicy, I'll just add in case of the resource to you.
Yes.
That New State University Computer Science program accepts clients.
Amazing.
For their students to do a task just like this.
Sure.
I've been this is my second semester as a client. And so they're doing things for me, like last semester they counted vascular bundles, sections of alfalfa, using object identification.
Amazing.
They did segmentation based on histological staining that I did. This semester we have 2 projects, so they've been great to work with. They can give you a data set at the end if that's like you write out your objective.
Yeah.
So if you want the contact for them.
Oh, absolutely. That sounds awesome.
They need clients every semester, so.
Cool. Yeah. I'll
We
connect
could do it
with
for
you.
free.
Yeah, my name is Denise Schiffner. I'm currently a researcher in the Smith Lab. I'm also a PhD student within APS focusing on the agroecology agronomy track. And then also I'm an MGS student. It's just basically a way for me to learn a bunch of cool ways to analyze data in spatial contexts. So yeah, that's fun. And I'm here presenting on predicting winter barley survival using the winter malting barley trial and weather data. So I did kind of think that Kevin would go before me so that he would explain what the winter malting barley trial was, but that's okay. I can explain it here too. Here's a map showing all of the different winter malting barley trial locations. The numbers next to the points indicate the number of years that they were measured for winter survival. And then the color of the points actually indicates the amount of winter survival they had averaged over all the years that they've been studying, or that they've been studied. There are about 20 different families that go in, Kevin. That's
Each year, 20
year.
to 25 entries that go into each trial.
and what's fun is they're not the same exact 20 to 25 entries, but there are some consistent ones across years so we can compare. But next, let's just focus in on St. Paul, Minnesota, where we have 14 years of weather data, or 14 years of winter survival data. And we also, what's also nice about this location is we have an intermediate amount of survival. So we don't have 100% and we don't have 0%. We have around 30 to 40%. So that kind of gives us an idea of what types of freezing conditions lead to winter barley survival or lack thereof. In St. Paul, we have the UMN St. Paul Weather Station, and that takes daily measurements on minimum and maximum air temperature, precipitation, snowfall, snow depth. And then what's really nice is it also takes information on soil temperature data. And it has two different conditions for that soil temperature data. Under bare soil, it measures at 4-inch, 8-inch, 20-inch, and 40-inch depths. And then under sod, it measures at all of those depths, and then also under 2-inch depth. Just to give a context, in the case of winter barley, John Hill Price was a previous postdoc in the Smith Lab. And while he was working with Kevin and Waleed, he published this paper where he planted winter barley and spring barley together at 3 centimeter depth in a greenhouse, and then measured crown depth at early tillering once the plants got to that stage. And he found that winter barley crown depth was at about 2.2 centimeters, or about 0.9 inches below ground. And the graph here on the right just shows the comparison between spring barley and winter barley, and then crown depth on the y-axis. The weather measurements that are interesting probably for us in that case are then to look at minimum and maximum. And in our case, we'll just look at minimum because we want to look at winter survival conditions under bare soil and 4 inches down, and then also under sod, 2 inches and 4 inches down, to kind of compare them to see what is likely most representing winter barley growing at that 2-inch kind of crown depth, or inch kind of crown depth. So this is kind of a busy figure. I'll lead you guys through it. On the left side, we have temperature on the y-axis, and then on the x-axis, we have date. So this is kind of the conditions that the winter barley was growing under from the fall of 2020 to the spring of 2021 in May. The red line that's going up and down pretty drastically is the minimum air temperature for that time. And then it's really hard to see, but of course there's a lag. There's 2 green lines and a brown line in here that kind of intersect and then deviate away from each other in some cases. The darker green line is associated with the minimum soil temperature 4 inches under sod. The lighter green line is associated with the minimum soil temp 2 inches under sod, and then the brown line is associated with the minimum soil temperatures 4 inches under bare soil. So looking at these conditions, we can see that the sod conditions kind of like, they correlate with each other pretty well. They're meeting up and honestly pretty close, even though there's a 2-inch difference in depth. But you can see this brown line deviates from them, being more affected by the air temperatures in the case where there is no snow on the ground, but then being more blanketed by the snow when it is present compared to the sod. In this case, we believe that representing kind of what the winter barley crown conditions are experiencing would be best represented by the minimum air temperature, snow depth, and then also that minimum temperature under bare soil at 4 inches. So now I'll go through some examples and show you the weather conditions in variable survival years for winter barley, going through first winter conditions in a low survival year, then in a medium survival year, and then in a high survival year. And then, excitingly enough, I pulled data from last week. So, right, we have data that we can speculate, like, what do you guys think that the barley is going to do this year? I guess we'll see. So, in this case, this is the same figure as it was before. We're just missing the sod measurements. So we still have minimum air temperature graphed here in red, and then the snow depth in blue, and that kind of minimum soil temperature here in brown. And this is actually pertaining to the field that Kevin showed at first, the 24-25 survival field, where we had a 0% average of survival. And you can see that the soil conditions first had a very significant drop in soil temperature at around this area, which doesn't allow for winter barley acclimation in any sort of way to allow for winter survival. But then it was also super affected by air temperature because it didn't have the blanket of consistent snow depth over those times where it would otherwise be mitigating the freezing effect. So we think that's why that's why that field just didn't survive, because all the plants froze. In the case of a medium survival here showing the fall of 2021 into the spring of 2022, we had about 59% average of winter barley survival, and we did have more consistent snow depth to mitigate the effect of that minimum air temperature. But we still had this dramatic decrease in soil temperature from basically, it looks like over almost like a day or 3, where the plants weren't able to acclimate. And then once they were at this temperature, they were kind of hovering at around 30, which is all right. Like, the plants can still survive at that point, but I think that significant drop off at first kind of contributed to a lot of the winter kill.
Hey, Sydney, sorry to interrupt.
Yeah.
Can you go back one slide though?
Yep.
Because while you have this here, it's more efficient. What do you think of like how warm it was though? In October, November, it's like warm all the way till like December, kind of.
Yeah,
Do you
definitely.
think like, did that cause an issue though too?
Potentially. In the past, there have been some planting date studies. I think Becky's on back in the day, and they did find that if you planted too early with winter barley, the plants could get too developed, and they wouldn't acclimate to winter necessarily as well. So that could have been a contributing factor. But I think more of the effect is this lack of snow depth and the fact that the soils got to, I mean, at the lowest temperature, 15 degrees. Like, that's pretty cold. That'll freeze plant tissue. So. But yeah, it could definitely be like a both kind of factor too.
And the other thing that can happen during warm falls is if you get, diseases or barley yellow dwarf or powdery mildew kind of hit those plants in the fall, that probably compromises them to some extent for their ability to survive a winter too. So yeah, we do need to pay attention to what's happening into the fall before the cold weather hits as well.
Having larger plants, like, benefit though, so I'm just thinking of like the counterbalance. If you plant earlier, presumably you're gonna have maybe larger crowns and maybe deeper crowns, or is that not the case?
I mean, that's what I would have guessed, but then we've had years where we planted, pretty late and maybe only had 2 or 3 leaves and we got good survival.
Okay.
So,
It's not.
I mean, I think that's something that we should explore more empirically. This is all just anecdotal, but yeah.
When does plural meristem tissue begin to develop? Because like we saw like with our facultative line, we had floral meristem tissue develop that same fall. In the fall.
In the fall.
Yeah.
Yeah, these, I mean, so.
The year where it survived.
Well, it kind of survived.
So most of our germplasm is photoperiod sensitive, so it needs to have long days before it will initiate reproductive state. But that's if there are lines that are not photoperiod sensitive, and that is a danger, that they would start to go into reproductive state before the cold weather hits, and then they're very sensitive to winter kill.
And then on to the most, well, not one of the most successful years for barley survival, fall 2019 going into 2020. And like Matt mentioned too, we didn't necessarily have the super warm growing conditions like we did in the low survival years. So plants maybe weren't as big going into the winter, but there was also a fair amount of snow depth, and that kept this kind of like acclimation temperature pretty consistent at around, it looks like around 32. So even though that's at freezing, the plants, as long as they can kind of acclimate to those temperatures and don't have these kind of crazy swings in temperature, it seems like they survive pretty well.
There was that pretty deep pre-snow, but maybe that's after the leaves, but the crown survived, or
apparently.
I mean, I suspect as we start to try to model these things, it's going to be something like the number of consecutive days below some temperature or something like that. We'll have to explore that whole space. Yeah.
It's a short drought too.
Yeah.
Like, the soil doesn't have enough time to
Absorb it,
Yeah. Are you thinking of doing some kind of a seeding date study so that you know you can kind of quantify how developed?
We've done that.
Oh.
And we probably can go back to some of that data and see if we get the environmental data from those years too. To look at that.
Yeah, definitely. Yeah, that's something else we plan on looking at too, is like the age of the plant or the stage of the plant going into the winter. Yeah, we're hoping to model that using these like winter weather kind of variables. What's your question?
Is the photoperiod sensitivity binary? Like it's 100% like sensitive or not sensitive?
Pretty much.
Okay.
Yeah, we don't see anything. I mean, even in these really warm falls, we don't see anything that starts to go into flowering, except our spring checks. So we see those go. Yeah.
Sorry. Is there a minimum amount of growth that you would need pre-winter for those fertilization cues to actually kick in?
Good question. I mean, all I know is anecdotally we've had plants that are pretty minimal growth, like I said, 2 or 3 leaves. And, probably wasn't the coldest winter ever, but they survived the winter just fine.
And I think most of these varieties are facultative too, so they don't necessarily have a vernalization requirement.
Right.
Yeah.
True. We have true winter checks, but the breeding program, our strategy is to do facultatives that don't require fertilization.
So, like I mentioned, this is measuring soil temperature at about 4 inches below soil, but can we predict soil temperature at winter barley crown depth or at around? Like I said, St. Paul measures it at 4 inches below bare soil, but due to John Hill's earlier study, we found that winter barley crowns are at about 1 inch below bare soil. So can we model and predict what that soil measurement or what that soil temperature is at around the 2-inch depth using just air temperature and snow depth? We also have other options. We have implemented these soil data loggers at 4 different winter barley trial locations for this fall, and then I believe we're also going to do it for next fall into the spring. This is at Crookston, Rochester, Rosemount, and St. Paul. And at those locations, we're measuring soil temperature, soil moisture and temperature at 2 different depths, that kind of 5 centimeter depth and the 10 centimeter depth. So we're hoping that we can use these measurements to correlate the 10 centimeter depths and then kind of accommodate or like have some sort of conversion factor between those two to kind of model what the soil temperature is like at that shallower depth and therefore simulate what's happening at the actual depth of that winter barley crown. Awesome. But okay, cool. That's for Minnesota. What are we going to do for everywhere else? So there is existing NOAA weather station data through the Global Historical Climatology Network, daily data. It contains records from 100,000 different stations in 180 countries and territories. And this network provides daily data on minimum and maximum temperature, air temperature, precipitation, snowfall and snow depth, soil temperature, and then some weird things too, like cloudiness, wind speed. A lot of these weather stations are used to predict the weather and have a lot of information, but they're not necessarily measuring everything all the time. So the majority of these stations have daily weather data on precipitation, snowfall, and snow depth. It's really interesting. I think NOAA really set up these weather stations to predict more like rainfall and snowfall as like those kind of metrics. And then most stations, actually less than the precipitation, have daily data on minimum and maximum temperature, but then very few stations have daily data on minimum and maximum soil temperatures, which is of course the data that we want. So just kind of give you an idea of what this looks like. I mapped in ArcGIS what it looks like. This is the availability of snow depth data, which is very surprising. I was like, oh, they're not measuring snow depth that many places. They are actually. This is data from 2010 through 2026 in the GHCND data, and there are 42,061 weather stations worldwide that are measuring this data, at least in that time frame. And this is the air temperature data, also relatively high resolution in terms of like us being able to find a weather station nearby that's likely representing what's happening on the ground. And there are about 20,000 of these weather stations available worldwide. And I have actually, they're very hard to see, but the WMBT locations are here in blue, or in yellow. Okay, great. But then we get to the soil temperature data. And for some reason, Oklahoma's on, but everywhere else is just very sparse. The WMBT locations are again in yellow, and the brown locations are the locations where they're either measuring 5 or 10 centimeter depth below bare soil, and then the green ones are 5 or 10 centimeter depth below sod. So I think we'll have to do a lot of calculations and kind of model building to see what that soil temperature, and obviously like the moisture and age of plants going into winter and all these kind of factors, how they influence winter survival. And like I said before too, there's only less than 400 worldwide that are measuring these metrics from 2010 to 2026. So,
one thing for the 0 to the 5 and 10 centimeter, depending on how they're measuring it too, like if it's just with like TDR probes, those data might be a lot noisier because it's at the shallower depths, it's a lot easier for like gophers to dig them up and things like that.
Sure.
So you might just like wade into that data set with some caution.
It definitely. I mean, even when I've pulled the data from these data sets, they'll say, oh yeah, it's available from 2010 to 2015 or whatever. And I'll see that all of 2013 is NAs. So it's like, oh, okay, well, it's not, but that's okay. Like, so there's even more like weeding through the data that has to be done and those types of things too. But there are also other resources that we can use. So this is just a list kind of that I've compiled of just a few resources that have this kind of climate data available. I think the GHCND is probably going to be the most powerful because it has most of the metrics we need, including temperature, precipitation, snowfall, and then those kind of sporadic soil temperature measurements. And then we have a few grid type data sets that are either produced by NOAA or NASA or kind of these like state agencies, but a lot of what they have is just temperature and precipitation. So trying to adapt those to replicate what's kind of happening at the Barley level might be a little difficult, but it may be useful for map generation. And then there are also these kind of smaller scale type data sets like the Midwest Regional Climate Center. They have a program that measures some soil temperatures in some areas, and maybe just utilizing those types of data sets and figuring out what's available will also help to kind of populate that soil temperature map to be a little more popular, or just modeling, of course, too. So on to the modeling side of things, we can do soil temperature predictions, and there are other people who have developed fancy models in order to be able to do these things. Like one, for example, is by Zing et al. And they found that using just air temperature and day of year, or even adding in solar radiation, can result in a model with an R-squared of like 0.94, 0.95, which is pretty impressive. I think in this paper, they were looking at more temperate areas, and they weren't necessarily looking over the winter. So I think snow depth has a huge effect on the soil temperature during the winter time, of course, too. So we might look at other models that are factoring in those things or factor them in ourselves as well. And then just like Maicy was talking about as well, kind of using these existing LT50 prediction models that exist for winter cereals, but maybe not for the winter barley accessions that we're looking at. Putting in all these fancy inputs and outputting a nice daily LT50 for us to get an idea of what conditions can these barley plants die under or survive under, or et cetera, et cetera. And then performing these freezing degree day calculations as well as a kind of companion to that, to kind of see what, for general in barley, what freezing degree day conditions do they need to be under in order to survive or not survive. And then once we have these metrics, hopefully we'll be able to generate a map similar to the one here. So this is one that was developed using machine learning with climate data that was looking at minimum temperature, maximum temperature, and precipitation, and saying what regions are ideal to grow corn in based on climate data. So we're hoping, and this is on the county level, but we're hoping that we can develop a similar map saying, what's your chances of winter barley surviving? Or like, what are the ideal conditions maybe for winter barley survival given the past, who knows, 10 or 20 years of climate data?
I've got another follow-up on the photo period. Has anyone done the critical day length of your facultative lines?
No.
Because, yeah, I think like September 15th is a 15-hour day still. So depending on like that, like maybe you're still getting, even in September, you're still getting a long enough day to initiate.
Yeah, and I don't know how old the plants have to be to sense that too, because we're planting at like the end of September and sometimes even into the beginning of October. So, but yeah, I think that's a good point.
I mean, my peas, they don't have the fertilization requirement, but they are water sensitive. And I plant with their young, but they don't, they also have to have enough growing.
Oh, I should have asked, are they long day sensitive then or short day?
They need long days to flower.
They need long days to flower. Okay. Yeah. So 15 hours, maybe by the end of September, it's like 14 and a half hours or something.
But I think they also sense.
The need for the soil temperature, I think with that network that exists, that extension data could be a source, at least for those locations where the network is located.
Yeah,
that
definitely.
would probably be a lot of reformatting and formatting data different.
Yeah, but I mean, it would be additional data, right? Like, I think I'm going to try to explore all of the possible kind of avenues that we can get this data. A lot of these winter malting barley trial locations are at research stations or are at like places where in theory they should be measuring these things, and maybe they just don't share their data with like the NOAA kind of outfit, right? So it's like maybe getting in contact with each of those individual stations and asking for, hey, like, do you have soil temperature data for X, Y, and Z kind of thing. I mean, it's logistically rough, but I think it's more informational than trying to like guess when we don't need to necessarily.
I thought I was going to talk a little bit about breeding progress. I mean, obviously last year wasn't a great year for collecting new data on our breeding program material performance. So thought maybe I would sort of step back a little bit and just talk very broadly about what we're doing in our breeding program and hopefully make some sense of it in terms of how it relates specifically to the winter that we're doing. So this is my attempt at trying to show what the broad spectrum of things we're working on. So when I started here, I was hired to work on spring 6-row malting barley. That's what Don Rasmussen made his career on over 43 years. And that's a covered barley as opposed to naked. So we also do some work with naked barley or polis barley. It was all 6-row, almost all covered. We had a tiny little bit of work that we did on naked barley. Actually, we have a line, it's not a released line, but it's a line that's available by MTA called M. Luna, which is a naked spring 6-row barley. And I'll mention that in a few minutes too. And then we have some other varieties that we released during that time. Then there was a barley cap project years ago. I can't remember what years it were. I think it was 2006 or 7 it started. And that was really sort of jump-starting some of the approaches to using GWAS and genomic selection in barley breeding programs nationally in the US. And that was about when the transition was starting to happen to 2-row. So that project allowed me to pull data from lots of different public 2-row breeding programs together into a shared database so that we could start a 2-row program. So that's kind of when we started the genesis of our 2-row spring breeding program. And I'm going to say a little bit more about that because that's kind of a full pipeline that I think will illustrate what we hope can happen on the winter side. And so we have a line, and I'll talk about that, called S2M184, which is an advanced breeding line that's a variety candidate coming out of that program. Then there was this follow-up to the barley cap was the USDA T-cap or Trinicy cap. And that was really the initiation of, or the big influx of funding that started a winter program. Again, this was a collaborative program nationally. There were several other breeding programs that were doing winter barley, but it was when we started our winter barley program and actually started to develop some genomic selection prediction tools to predict winter survival, which is a very important tool because as we've seen already, actual winters aren't always the best way to screen for winter survival. So that's kind of when we started our 6-row program in facultative or winter barley. And we did release a variety of M&E equinox that came out of that program. Of course, the industry wants all 2-row barley now instead of 6 rows. So that kind of caused us to pivot again and try to work on developing 2 rows more. There's also another project that we're involved with. It's a USDA OREI project that's now in its 3rd iteration. So we've been at it for almost 8 years now, developing multi-use naked barley for organic production systems, but more or less in general as well. And that allowed us to really just start work looking at both spring and winter naked barleys. So we've done, I won't talk about the details, but we've done some really interesting work with naked barley as to the different kinds of value that it can provide, both in terms of malting, brewing, distilling. We've had some collaborators work with naked barleys that have found some really interesting applications in the end uses. And then in the work that we've done working on Fusarium head blight, which is a really critical disease for small grains in the Midwest and actually all over the US where small grains are grown, we found that you can mitigate a lot of the risk of the toxin produced by the Fusarium pathogen, deoxyribonucleic, with naked barley, because a lot of the toxin accumulates in the hull. So kind of looking at all of these different aspects of or sort of applications of barley in both winter and spring, and focusing more really on 2-row. So that's kind of the big sort of backdrop. And of course, what we're really interested now is how do we develop a 2-row winter barley? And that's, I'll, at the end of my little presentation, I'll talk about 2 advanced breeding lines that are part of that program that we've been developing. But what I wanted to do is just kind of walk you through the development of the spring line so you can see how our breeding pipeline works. Yes, and be a little bit depressed at the end, I think. But so we start with a crossing block. You know, we make lots of, we probably make, in each of these different programs, we probably have 50 different crosses every fall. And we all do all of that work in the greenhouse. We grow up the F1s in the greenhouse. We actually do single head selections in F2s in the field. And so that all happens in that first year. So we're able to do 3 generations a year if we're using greenhouses. Then we grow up our F3s in container racks. So we can get about 50 individual plants in a rack. So we can jam quite a few of these into a greenhouse. Again, in the fall, we go off to a winter nursery in New Zealand. This is what we used to do. Things always change a little bit every year. I haven't been in New Zealand the last couple of years, but this is what we did for this particular line. And a lot of the surrounding fields were quite interesting because we have a lot of sheep. But we have, like here, what you can see are all of these head rows. Each row, little 5-foot row, is a different breeding line in our program. And so we do a seed increase in New Zealand so that we can harvest the selected rows from that nursery, get that seed back to Minnesota in time to get it planted in the spring. And then what we do is we plant these yield trials. They look something like this. We start out with 3 locations with our first year yield trials at Lamberton, St. Paul, and Brookston. Then we go to second years, which are essentially the same locations, but more replication. And then eventually to advanced breeding programs that are more statewide. And then we go in like 11 different locations when we get to these statewide trials. So, you know, we get quite a bit of data over the years on these lines. And then of course, each stage of the program, we're doing selection, we're reducing the number of lines down. So our first year trials, we might have 300 entries. By the time we get into the state trials, we might have 5 or 10. And then at that point, we also start to do quality evaluation with the industry. And this is really one of the most challenging parts of doing breeding for malting barley is trying to meet the very rigorous parameters that the industry has for successful malting barley. So barley production in the US, generally, if you're producing malting barley, you are almost always contracting with a malt company or a brewery, and that contract usually specifies what variety you're growing. So there's a limited number of varieties that are accepted by the brewing industry. And this American Malting Barley Association is a consortium of maltsters and brewers that has an evaluation program to evaluate the top candidates from all of the different breeding programs. And then they have a technical committee that evaluates whether they meet those criteria or not. And if they get through that gauntlet, they might get put on what's called the recommended list, or AMBA. And then those are the varieties that would usually end up in contracts for farmers to grow. So this is the email I look forward to every spring. So you put about 4 or 5 entries, your best lines, into this industry program, and you get this email back in April. Haven't gotten this year's back yet. That says either satisfactory or unsatisfactory. And this is in the pilot stage, and usually you have to get through the pilot twice. So you have to get satisfactory 2 years in a row, typically, before it gets advanced beyond that. And then you get some nice comments about what they liked or didn't like about it. And so this line S2M184 was rated satisfactory 2 years in a row. So it made it through that, which is we haven't had one happen for quite a few years that got through that. So based on that, then we did a large 40-acre increase near Crookston with a farmer in anticipation of there being a large plant scale evaluation. Usually what that entails is one of the large malt companies will contract like 300 acres or maybe a couple of 300-acre fields to produce enough to do a plant scale actual malting. And then that malt gets distributed to commercial brewers that use it at a commercial scale. And then they actually make beer out of it for the first time in this whole process and can evaluate the beer. So we did this big seed increase anticipating, because we'd also been working with a large malt company that seemed to be interested, but they wanted to do their own evaluation. And so they did 3 years of evaluation after all of this to see if it was really going to make it in their particular malting applications. So they did that for 23, 24, 25. They sent us data. The data all looked pretty good for those first 3 years. And then I just got this email, which says, well, we decided we're not going to look at it anymore. So they suspended the evaluation, they declined it, but they did say, oh, but we do like this S2M204 and 207, so let's keep looking at those. Of course, they've only looked at them 1 year. So anyway, my point is to show you that there's this fairly long path. If this is the normal path that we have followed for developing malt and barleys, and in my own humble opinion, it's a little bit broken, and I think there's some alternatives we can come up with. But I show you this to show you what we would expect on the winter side. So remember, this is all a spring barley. So in our winter program, we now have these 2 lines, which the winter ones all start with a W, W2M004 and M005, that are both in the first year of this pilot testing for AMBA. So we'll hopefully again get data probably this April on how those 2 lines have done. So that's kind of where we're at in that process. I'm thinking I have some data. I'm probably not going to spend a lot of time on looking at the data, but these are just some tables with those 2 lines bolded. The checks are sort of above the line, and because we have a facultative breeding program, we can evaluate these lines in both spring planted and fall planted trials. So that's why you see different columns for spring planted and fall planted. It's one of the main reasons I wanted a facultative program, is because when everything dies in the winter, you can still plant some things in the spring and get some useful information out of those trials. So, I'll just point out a couple of things. Look at M004. It has not quite the winter survival compared to Equinox. And then note, this is like 19 different experiments where we've done winter survival. So quite a bit of data, but still pretty reasonable winter survival over those 19 experiments. Yields were better than Equinox, so that's a good thing in the fall, but in the spring, not so much. And so this is one thing that we've noticed, is that many lines perform similarly in spring and fall, or they rank similarly in spring and fall, but some are quite radically different. And so this is an example of that, where if you look at the heading dates, it's kind of normal-ish for heading, but it's actually quite late when you plant it in the spring. So there's probably some sort of photoperiod thing happening there that's segregating in our breeding program. Again, it doesn't affect everything. But one of the big problems with equinox was lodging and stem breakage. It tends to stir breakdown early. So our advice to farmers is when it's ripe, harvest. Don't wait, because it will tell you when it's ready to harvest, when it starts to fall over. Anyway, so that's one of the things that we've improved very generally in the program is much better lodging and stem breakage than equinox. So that's some agronomics. There's some disease things. I don't know what to sort of point out, maybe a couple of things. Net blotch was a little bit high in equinox, and it's quite a bit better in the newer lines that are coming out of the program. Powdery mildew is something that we typically have not even screened for in our program. It's not usually a big problem in spring barley. It is a problem when you have both spring and winter barley because you get this bridge effect where you get winter barley getting infected with powdery mildew, and then the spring barley gets infected with the inoculum that comes off of the winter barley. So it's now a disease that we're looking at a little bit more closely. Didn't really mention much. I think last year we talked about, we've done some experiments looking at Fusarium head blight and Don, and we think that winter barley will have an advantage in general because of the shift in the phenology. So the development of the plant is 2 to 3 weeks earlier than spring barley. And we think that that could be a mechanism to actually avoid the critical periods when Fusarium is at high risk. And there's different lines of evidence to show that actually may be the case. Obviously, there's genetic resistance as well, but this would be sort of an avoidance. And then here's some quality data. So this M004 looks really pretty promising. Green's obviously better, red's not as good. Some traits, whether it's good or bad, depends on what the actual brewing procedure is. So there's a difference between doing adjunct brewing, like some of the big brewers do, where they add corn or rice into the grist, versus all malt brewing, which is what a lot of the craft brewers do. And that has to do with these parameters called diastatic power and fan. So you typically want those to be lower in an all malt beer and higher in an adjunct type beer. So this 1004, it looks pretty interesting to us because it has more of that craft profile, has low protein, all the things that the brewers are looking for. So that was one of the reasons why we've added into that AMA trial. And then this is also quality data, but from fall planted trials. So one thing that's been encouraging, as we look at data from these fall planted and spring planted trials, is that the quality parameters tend to rank pretty similarly between fall and spring planted trials. Even though some of the agronomic parameters aren't necessarily ranked the same for alternate types. So that's nice because quality data is very expensive to collect. If we get a severe winter, we can get some decent quality data from our spring planted trials as well. So I didn't really dive into the data too much, but suffice it to say that we have a couple of lines that are starting with this industry program. We've got a full pipeline of things behind them. So as onerous and potentially disappointing as that path to commercialization is, there are things in that pipeline. I was just going to finish up with, I just threw some pictures up here with some ideas about alternative paths to commercialization. So, I think this AMBA quality program is a path. I think it's probably something that we need to explore alternatives to. So very recently, we've been working with a small malt company, Vertical Malt, a small distiller, Black Frost, with this MN Luna, which is not a winter barley, it's a hulless barley. And we've got it malted sort of at a commercial scale. We've got it distributed to some local brewers, have given us some positive feedback. Black Frost Distilling is in New Ulm, and that particular distiller was really excited about the possibility of what a hulless, in this case, what a hulless barley would do. And, is particularly interested in like a single malt, sort of an American single malt that could be made with it. So the idea is that, rather than this big industry pipeline where we're trying to get it through this gauntlet of large brewers and maltsters, what can we do locally with the folks that are around here to stimulate some interest, some local interest, enough so that somebody might license this variety and grow some limited acres for seed, because we can't, I can't be a seed producer. I mean, we only have a limited amount of seed, and what will happen often is people say, "Oh, that looks really promising. Can you send us, like 100 bushels?" Like, well, no, I can't. So we need to find a way to find some local seed producers and local maltsters and local brewers that have sort of a mini version of maybe AMA. So also working with the Artisan Grain Collaborative, and that's really very well aligned with their mission, is developing a local grain shed. So they're pretty excited about that. Right now they're working on ways for me to find maltsters and brewers to take, and then Luna, again, this is a spring naked barley, but we're going to grow it because some of the brewers that are interested are organic brewers. We're going to grow some of that at Morris on organic land. They have a large organic tract now at Morris. Enough that we can do some small scale kind of commercial testing of it. And then this other picture is this event called Farm to Flavor, which is part of this culinary breeding network that has an annual event every year to sort of link breeders with culinary folks of various types. So lots of different crops and then different chefs, makers, brewers that make things out of this. And so we presented with 2 brewers from the Madison area, Starkweather and Irby area, that were with us, that made a beer with this MN Luna featured at this showcase. And both of those brewers were interested in using more of it if there was larger amounts of it available to actually use. So that really becomes kind of this challenge and something that is a little bit outside of the scope of a plant breeding program, but still somebody has to do it, I think, to get things going. My thought is that if you have at least a small scale local use of something and somebody's producing the seed, then if I get inquiries or people want to try it, there's at least a seed producer to go to rather than me saying, well, I can give you like a pound of seed and you gotta increase it on your own or whatever. So anyway, it's kind of a work in progress, and I think Artisan Grain Collaborative is really quite interested in helping out with that as well. And then of course I've been working with our own FGI commercialization folks with large maltsters to look at the winter barley side, but these guys are equally interested in spring, winter, whatever. So I will stop just so we have a few minutes for questions. Folks have any questions?
Typically malting barley uses the holes, right?
Yes.
And so what's the drive to use naked barley instead?
So yeah, so the hulls are really important in this lautering step, which is a filtering step. So it forms like a filter bed and then the wort is filtered through those hulls. If you don't, so you can brew with other things like wheat, wheat does the hull too. You need to either have a synthetic filter, a mash filter, or you need to add like rice hulls or something like that as a filtering mechanism. So people do that if they want novel ingredients. The brewers that were interested in trying naked or hulless barley initially were interested because of the beer yield. So you don't make a lot of sugars from the hull. So in theory, you have a higher malt extract from a naked barley, which it should give you more beer per gram or per pound of malt. But I think there's also some flavor components that would be interesting. The hull often can impart some off flavors. So reducing that hull might help reduce those off flavors. And then the brewers, we've done some brewing trials with this naked barley and you can blend it. So you can do like a 50-50 blend of covered in naked barley, and you don't need to use a mash filter or add anything else. So there's some ways to kind of play around with recipes to see if that might be useful.
And the focus of going from 6 to 2 row is mainly just the size consistency between the kernels, or
that's the industry really went in that direction. 2-row kernels generally have a rounder shape, and so in the malting phase, you take up water. And so there's a more even uptake of water in the malting phase than in 6-row kernels, which are a little bit, different shape. There are other things too. There's sort of breeding histories. They're very different breeding histories. So they have other things that are different that actually have nothing to do with the spike shape. And a lot of this is sort of tradition.
I misspoke before. It's 12.5 hours on September 15th. I was thinking May 15th. So, so just throwing that out there. But yeah, it'd still be interesting to know the critical day length though.
Yeah. We have not measured that, but I think you're right. It would be good to do that. Yeah.
What does crossing planning look like? There's so many things to breed for. Do you plan crosses at all, or is it just
Yeah, no, we just. Yeah, so it is, it's challenging because there's a lot. So we have crosses that we think are going to possibly result in a variety, and those are usually the sort of the elite by elite crosses where, you fixed a lot of the traits in common between the two parents. And then we have more parent building type crosses where like beta glucan has been a problem. Beta glucan, if it's too high, the brewers don't want it. So getting that down. And so initially we had to do a lot of crosses between things that had low beta glucan, but we had to fix other things.
Yeah.
So it's a combination. You have to kind of look at sort of line up all of the positives and negatives and try to complement each other as much as you can. I mean, we've done a little bit with like using PopFar, doing simulations to simulate parent combinations. We use it to some extent, but I haven't really validated it because you make the cross based on those predictions, and then like 5 years later, you're supposed to ask, but it didn't really help. I haven't really gone back and look. So
In the past, you've talked about your magic population for winter hardiness.
For winter hardiness, yes.
Just curious how that project is going and how those lines performed last.
Well, that one survivor there was in the magic population. So there were a couple. And then unfortunately we did not plant it in Rosemont. It would have been a perfect year to plant it in Rosemont, but it was planted in a couple of those. So we have a few more locations. We'll probably publish that once all of that data. It's in the field this year as well. So I think we'll see what happens when we get out of this year, and we'll publish that data. We've already taken lines from the magic population and put them into our breeding program though. So we're crossing to them. What we'll hopefully be able to do is use the magic population as a training population to do predictions in the progeny from those crosses to help kind of accelerate selection for winter harding.
What was the magic population?
So this was a population that Brian Stephenson's group created, and so he did a ton of germplasm screening for winter survival and picked, and there were two very large GWAS studies that were done, and so we took the phenotype screening that he did, the two GWAS results, all the favorable yields for the QTL from those results, and then picked a set of 8 parents that were, phenotypically winter hardy and carried all the favorable alleles for the GWAS hits in these 2 big GWAS studies. And then use those as parents and then did the typical sort of process to develop a population. And it's about, I think it's under 700, like 680 lines. And it's shown nice segregation for, and the early GWAS results, picked up some of the hits of things that we would expect. There are some genes that have been identified before, but also novel ones as well. And then we haven't used it to do sort of predictions yet, but I think that's the next step.
Last thing I wanted to say is I like this approach to just building commercial interest at a more local level that we did. Those 2 winters that are in the AMBA program, they drop out at one of those many, veto points. Then, but you still think they have potential. And I know you're in touch with that commercialization team a lot, but you can help reduce the pilot volume. So there are a few hundred pounds sitting there waiting for do some outreach, and it seems like a deep process.
Yeah, that's exactly what I was thinking. Even this 184, I'm going to reach out to some folks and say, look, the big guys have passed on this. If you want something that's locally adapted, it does well in Minnesota. It's also a GN minus, so it doesn't produce glycosidic nitrile, which is a really important thing for distillers. So, and that's who's been using it as Black Frost. Has been using the naked line, but they would very much like a GN minus line. So yeah, anything that gets sort of kicked out of the AMBA thing, that's fair game, and we've already probably increased it to a point where we have stuff available to that right away. So absolutely, yeah, or maybe not even wait for it.
They're not on the rights. Do they take rights or like
So they have some funny wording in the grant contract that gives them sort of first right of refusal, but they don't ever want to license anything. They just want to be at the table when it comes to that point, because their main thing is they don't want a university program that's been supported by this consortium to release a variety in a way that would benefit one member but not another. So if you licensed it to Anheuser-Busch or something like that, then the other partners would be kind of upset about that, right? Because they all contributed to that. So that's the main thing, is they want it to be So as long as you're publicly releasing it or doing a non-exclusive release and it's equally available, then they're fine with it typically.